This is a family saga.
An extended family relationship. We go a long way back.
700 or 800 million years back, when multicelled organisms first set off from our single-celled ancestors on the tree of life, something interesting happened. We never left bacteria behind.
We took them with us.
It was a partnership from day zero.
They've been living inside and on us for the entire ride up the evolutionary tree.
We depend upon them, and they depend upon us.
This wonderful cooperation where we both benefit.
Yet for most of our history, we didn't know they were there.
The first evidence of microbes came in the late 1600s. A Dutchman by the name of von Leeuwenhoek.
He built the first microscopes and described the first microbes.
An unbelievably great company of living animalcules, very prettily moving through water.
He was pretty enthralled with these microorganisms.
But wonder gave way to fear as scientists discovered that many of these tiny creatures had been making us sick.
These are bad organisms, truly horrible pathogens that have plagued human existence.
Which led us to believe that eliminating them from our environment, from the water we drink and the food we eat, and from our bodies would usher in a new era of public health. And it did. Hygienic practices led to a spike in human lifespan. New antibiotics like penicillin became wonder drugs, saving hundreds of millions of lives. It seemed the only good bacterium was a dead bacterium.
Better shoot to kill.
But our ideas about germs are changing.
We are beginning to understand the very subtle roles that they play in regulating our own health.
They help us digest our food and fight off dangerous invaders.
We don't know whether a human would be able to live any meaningful existence in the absence of microbes.
Inside every one of us is an entire ecosystem, a microbiome. And scientists began noticing something else.
What led to this extension of human life and reduced human suffering also may have brought with it their own set of problems.
Many diseases are on the table.
Obesity, heart disease,
chronic inflammatory diseases.
Things like Crohn's disease, diabetes, and many others, all on the rise. Our war on microbes may have caused unintended collateral damage, our own health. So, getting back on good terms with our longtime fellow travelers could mean the rebirth of a beautiful friendship.
By the end of today's program, you may have a very different appreciation for the bugs in our environment, particularly bacteria, which is what we're talking about today. The first thing you need to understand is that they vastly outnumber us. Each one of us has about 30 trillion cells, human cells, that are uniquely ours. But did you know that living in and on us are 39 trillion bacteria? Okay, without further ado, let's introduce our participants. Our first participant is a professor of medicine, microbiology, and immunology at Stanford. He's a pioneer in the study of the human microbiome. Please welcome David Relman. Our next participant is director of the Center of the Microbiome Innovation at the University of California, San Diego. He's the author of Follow Your Gut and Dirt is Good. Please welcome Rob Knight. Our third participant is director of the Wisconsin Institute for Discovery at the University of Wisconsin-Madison. She was a science advisor to President Barack Obama. Please welcome Jo Handelsman. And rounding out this distinguished panel is Professor of Medicine and Microbiology at the New York University School of Medicine and the author of Missing Microbes. We'll talk about that in a bit. Please welcome Marty Blaser. David, let's start with you. So we evolved with microbes from the very beginning, and we just told everybody we all have our own little world living in and on us, but when do we acquire that? When do we get it?
So the beginning is a little bit uncertain. We forget that our own individual histories begin before birth. which means in this case that our own fetal development was influenced by our mother's microbes and her microbiome. We have probably a little bit of direct encounter with some of those microbes or parts of them before we're born, but then everything changes at the time of birth, and it's that passage through either the birth canal or out through the abdominal wall that causes a sudden change in our relationship with the microbial world.
And Rob, you have some interesting thoughts about this and a personal experience. Tell us a little bit about your daughter, correct? And what happened there?
Yeah, sure. So after our daughter was born, as you know, when you have kids, things don't always go according to plan, and that was true for us, and we wound up having to have an unplanned C-section for medical reasons. And straight after she was born, we coated her with the microbes that she would have had she come out the regular way.
So you feel that strongly that exposure in the birth canal is...
We felt that strongly as parents. Now, the AAP recommends that you not do this now that there's a lot of public interest in it until the research is done. So it's not something you should do from a clinical perspective right now. Of course, as a parent, you're probably going to have to do a lot of things that are not 100% based on evidence.
That's right. That's true. I can tell you from experience. You know, we saw in the brief piece a whole bunch of diseases that rolled by. There seems to be some suggestion that there's an autoimmune component. By that I mean that the microbiome somehow interacts with our immune system. Marty, can you talk about that? And just to set the stage here, how might these interactions be happening?
So mostly I talk about the microbes in the gut, and that's where a big part of the immune system is, in the wall of the intestine. And it's pretty clear that there's interaction between the microbes and the immune system and vice versa. And it's become more and more clear that the microbiome is training the immune system. It's teaching the immune system what are the good bugs, what are the bad bugs. And if that training is off, if it's misaligned, then there's a risk that the immune system won't develop in the proper manner. So that's what we think are driving some of the immunologic diseases.
Two-way street there.
Yes.
Before we go any further, we need to sort of set the stage for how a lot of the work in your area is done, and it's done with mice. Joe, can you explain a little bit about how you use the mice in this very unique way?
Well, the mice have been particularly powerful because they can be raised in a sterile environment, so they can be delivered by cesarean section and kept sterile in a bubble, and then they can be inoculated with any microbes you want. And so it's the only system we have where we can do a very clean mammalian microbiome or microbiome-free gut, and then add back one organism, a few organisms, or the entire microbiome from the mouse, or even another organism like a human.
And mice studies is this primarily the way most of the work is done?
Well, one of the powerful things in the microbiome field is that it brings together many kinds of data. It's epidemiological data on humans, looking at large populations and correlations, and then experiments on humans in situations where we can actually do the experiments with variables on humans, and then mice and other model systems play a major role.
Briefly back to humans, though. When we're first born, we acquire the beginnings of our microbiome. When does that stabilize? When do we say, you've got it, this is you, and how long does that take? Rob?
For the gut microbiome, it's about 2 to 3 years, and then for other sites like the skin and the mouth, it's a lot faster, so within a few months.
And yeah, go ahead.
I would just agree, the vast majority of what we see easily is established by about 2 or 3 years. I think we all would agree, though, that there have to be some other and really important but more subtle features that take a little bit longer, well into later childhood, maybe even at the time of adolescence and beyond.
And even through old age, so the first 3 years are where you have the most dramatic difference. So it's like your microbiome's going from seawater to soil. It's that profound a change compared to different physical environments. But then between the time when you're 3 and the time where you're 90, that's more like, say, the difference between one patch of farmland and another, rather than being a completely different kind of environment. So the change is still important, but it's subtle.
And this is the time frame when a lot of parents who have little kids know that they get ear infections and antibiotics are brought on board. Is this a good thing, a bad thing?
Well, I mean, it is what it is. Everything is a trade-off. In this particular case, antibiotics, as you've heard and seen, have had an amazingly important role in saving lives when they were used for, what turned out to be good purposes. But of course, they have collateral damage that they cause, and everything is a trade-off. So when they're not used for a good reason, those downsides are going to outweigh.
You have an opinion on this.
I do. So the point is, as we know, antibiotics are miraculous when they're used appropriately, but they are overused tremendously. The CDC estimated 30% of the antibiotics are overused. I think the actual number is higher. In Sweden, they're only using 40% of the antibiotics that we do. So it suggests that maybe 60% of the antibiotics aren't necessary. And in the US, there's tremendous variation between doctors, between regions, parts of the country that aren't explained by differences in disease. They're differences in practice and in culture. Some people and some doctors want to use antibiotics more, others don't. There doesn't seem to be much difference in outcome.
So let's talk a little bit about the Human Microbiome Project. A lot of people are familiar with the little petri dishes. That's what we think about when we talk about studying bacteria, but there's a whole new way to study bacteria. Joe, can you sort of walk us through what that is and how that intersects with the Human Microbiome Project?
Well, about 30 years ago, scientists began to realize that we'd been missing a vast amount of knowledge about the microbiome, every microbiome, not just in humans, but in soil and in oceans and on trees. And the reason that we've been missing that information is a lot of microbes, the vast majority, won't grow in culture. So those petri dishes that we all associate with microbiology are only one little sliver of the story of microbiology. And so the effort became to study microbes without necessarily culturing them, and to circumvent culturing them, we started extracting DNA. And so that's the major way that we describe microbiomes today, and most of the work we'll talk about here is based on DNA analysis.
So it's not the old-fashioned petri dish. That's gone.
Some of it's not gone at all. Some of it still is, and when we want to study a particular organism and it can be cultured, it's one of the most powerful tools we have. But if we want to look at the entire community, including the half, or in the case of other environments besides the human system, as many as 99% that can't be cultured, then we need to go to the DNA method.
And Rob, you've done a lot of work with this. Tell us about it, and let's take a look at some of what you've done, if you can describe it.
Sure. So what you're seeing here is my daughter's microbiome developing over the first 2.5 years of life. And what you can see is she's starting off with these different regions: the gut and brown, the mouth and blue, the skin and the mouth and green, the skin and blue. And she's moving more and more towards the healthy adult state, as defined by about 15,000 healthy people in the American Gut Project, which is our citizen science initiative where essentially anyone can get their microbiome sequenced. And so what's exciting about this is even to a 6-year-old, you can explain how she's starting off in one place on this map, she's moving to another place, and what the regions are that are defined by health and disease.
So explain again, the different colors represent?
The different colors are the microbiomes in different parts of the body. Each dot is the whole microbiome, so potentially 1,000 microbial species from one individual. And we place 2 dots on this map close together. If they're more similar in terms of the evolution of the microbes that they have in them, we place them further apart if they're more dissimilar in terms of that evolution.
How unique is each person's microbiome?
Every person's microbiome, every time you sample it, is unique. So if you sample yourself today 3 different times, and you sample yourself tomorrow 3 different times, we can tell those apart from each other. So it's all a matter of scale. It's about how different are you from yourself, how different are you from other people, and which of those differences are large and which are small. And it's important to remember that you don't have just one microbiome. The microbiome of your mouth, your gut, your skin, they're all completely different from one another.
In fact, Rob's gut is a lot more similar to my gut than my gut is to my mouth, for example.
Mm-hmm. Even within the human mouth, there's diversity. Is that correct?
Absolutely. A very striking demonstration by Julie Segre at the NIH about the different habitats of the human skin and how they're as different as Rod was saying, as grasslands are from forests are to deserts.
Let's talk about what we've been doing in the last, 40, 50, how long is the...
70 years.
70 years. That's when antibiotics first came on, about 70 years ago. And this is an area of intense interest to you, Marty. In fact, you wrote a book that's been compared to Silent Spring and its importance in terms of understanding exactly the impact we're having on our own ecology, if you will, called Missing Microbes. Tell us a little bit about that book, your thesis, and sort of what you think about the future.
Yeah. So the idea begins with many of the things that we've discussed before. The idea that the microbes that we have in the human body are ancient, that we've inherited them from our ancestors, that they're formed, the structure of our microbiome occurs early in life, that they have benefits to us, they're doing all kinds of good things. And now there's more and more evidence that our microbiome is changing, that we have lost diversity, that we have lost important ancestral microbes. And I hypothesized that this has consequence. And there's more and more evidence that the consequences are related to the diseases that are now becoming epidemics. Since World War II, we've had rises in asthma, in obesity, in diabetes, in juvenile diabetes, in inflammatory bowel disease. And there is an increasing body of evidence for all of these that the change in the microbiome is playing some role in the story.
You and I were talking beforehand, and you likened it to other sort of human intervention impacts like climate change.
Yeah, so climate change is how humans are affecting the macroecology through modernization, through all the things we did. When Henry Ford or Benz developed the internal combustion engine, they didn't anticipate that every time you turn the ignition that the ice cap in Greenland is going to melt a little, but we think that that's true. And in a sense, I think that that's what's happening in our human bodies. Every time we take an antibiotic, perhaps a few organisms become extinct.
And exactly, yeah, that's kind of profound.
Change in our microecology is paralleling
and
what we're doing in our macroecology.
You also said you think what's happening to our microbiome is happening much, much faster.
Yeah, well, we're 70 years into the antibiotic era, and antibiotics are not the only factor. C-section, there are many other factors involved, but maybe it's the most important. And so it's a difficult problem. Antibiotics are miraculous. They save lives, but we're overusing them. So we have to dial it back, and we have to understand what we're doing, and then how we can restore what we've lost.
Dirt's good, right? Title of your book. Tell us why dirt's good.
Dirt's good because exposure to a lot of natural microbes out in the environment, whether it's from dirt or whether it's from animals and farms or healthy water, healthy soil, and so forth, has a surprising number of beneficial effects and can give the microbial diversity that we need to reduce the frequency of some of these conditions. Although it's an open question how much we can restore. Like, I think a much more direct analogy with antibiotics is DDT, where it saved millions of lives, and yet you have this hugely deleterious effect on the ecosystem. And then afterwards, just introducing a few rats and pigeons isn't really going to cut it for improving biodiversity again, right? So understanding what we're changing and what we need to do to restore it is a really key scientific challenge at the moment.
In the meantime, if you're a parent and the pacifier hits the sidewalk, do you boil it? Do you call in the hazmat team? Yeah,
well, it depends a bit where the sidewalk is. Like, if you think there's a lot of lead, maybe you shouldn't. Maybe you should wash it off. But it's more a chemical than a microbiological thing that you should worry about. So if it drops on a hospital floor, you probably want to wash that off, right? But if it's your floor at home, or if you don't have any reason to believe that there's anything bad there, probably the positive effect outweighs the negative.
Any opinions here on the 6-second rule?
For some reason, NIH hasn't funded a lot of research on that.
Interesting. No. Big problem. Let's talk about some of the specific diseases that we've been alluding to. The first is asthma. Asthma is a major public health issue. If you're in New York City, there are certain neighborhoods in New York City that are truly plagued with asthma. Overall, 8% of Americans have asthma. But we've looked at these sort of ecological studies, and the story is actually very kind of stark when you look at very particular communities. Two of the communities in a recent article in the New England Journal of Medicine looked at the Amish and the Hutterites. Why those communities, and what did we learn from them?
Yeah, so this was work from Jack Gilbert and Erica von Mutius and a number of other of their collaborators. And what's amazing is that the Amish and the Hutterites both live what we would consider relatively traditional lifestyles, very farm-based, but they have completely different rates of asthma, where the Amish have essentially no asthma and the Hutterites have a lot. And what's different is that the Amish will take their kids on the farms to interact with the animals, whereas the Hutterites have a very tight distinction between farm life and home life, and they make sure that they change their clothes so they don't take their kids out on the farm and so on. And the exposure to particular kinds of microbes, including some kinds of lactobacillus, seems to be really important for that difference in health between the two communities.
The Amish were 6 times less likely to have asthma and allergies than the Hutterites. And the Hutterites have asthma rates that are consistent with the rest of the country. Does this go towards what they call the hygiene hypothesis for the development of diseases? Can anyone?
That's right. So the first iteration of the hygiene hypothesis was the idea that exposure to childhood pathogens was good. And there hasn't been a lot of evidence in favor of that. What we're thinking now is much more along the lines of its exposure to this diverse array of beneficial microbes from the environment that's good. But by trying too hard to keep yourself away from the pathogens, you may also be walling yourself off from those beneficial microbes.
David, did you want to add to that?
No, I mean, a lot of this, as Marty was saying, is just a function of what we have been doing both to ourselves and to our environment. It's a function and part of the built environment and how we have segregated ourselves from the environments we used to be born and raised in.
And it's striking that kids traditionally have liked to eat soil. And a lot of animals eat soil. In fact, when they're sick, some animals self-medicate with soil. So it seems like there's some natural process of ingesting soil, and maybe we're learning why that was an impulse for so many kids.
In some ways, when you look at those kids and what they do and what we all do, it's really also about continuing to sample our environment in order to find those missing microbes, right? That can fill in the niches and habitats that have been emptied. And so in some ways, that natural behavior is really a very adaptive one.
I know you guys are working on some additional information about this.
Yeah, we're working on a new study with Erica in Bavaria where you can go down a street and you'll have houses that have cows where they bring the cow into the barn versus houses that don't have cows. And even on that level, one house to the next down the street, if you have a cow, you're much less likely to have asthma than if you don't have a cow. Now, obviously in New York, recommending that every parent get a cow
creating a run on cows
not particularly helpful.
So this is an ongoing study that. Okay. All right. So that's asthma, but I think even more interesting than asthma is obesity, and we all know that obesity is an epidemic in this country. This is not news, but Marty, you've overlapped some information and what you're talking about in terms of the obesity epidemic and some of the.
So there are many things to say, but I don't want to take up the whole time. But one of the most important observations.
These are the rising obesity rates.
These are data from the CDC, and the darker the color, the higher the percentage of obesity. So you can see the difference between 1989 and 1999 and 2010. So this is only 21 years. Something very important has happened. It's happened all over the country. And in about 2012, we made the connection. I think it's in the next slide. That if you look at the obesity map and the regional variation in antibiotic use, there's a lot of similarity. There's a lot of overlap. So that's it. That's kind of interesting. It doesn't prove anything because it's observational. But 70 years ago, farmers found that if they gave antibiotics to their farm animals, it would change their growth. They would grow bigger, they'd grow faster. It changed their metabolism, and they found that the younger they gave the antibiotics, the more profound the effect. So we thought that maybe that's one of the things that's fueling this, and we began to do experiments in mice, like as with others, in which we could give antibiotics to mice as the only variable, and that increased their obesity.
Joe, you've done work on that. Can you take us through what these mouse experiments where?
Yeah, this isn't my own work, but other people have done obesity experiments where they transfer the microbes from obese humans into mice, and they see the mice gain weight quite dramatically. I think this was one of the turning points in the microbiome research field, because we began to see cause and effect. As Marty said, those maps show you great epidemiological similarities and correlations, but this was a direct effect by transferring microbes.
It's amazing. I mean, how could this work? Is anyone working on what is the mechanism, the elixir, the genetic turn that happens here?
What we and others are working on, it's not genetic, it's environmental. It's because the obesity epidemic is too fast to be a genetic problem. It's not taking multiple generations, it's happening in years. And again, it goes back to the early years of life when the microbiome is forming. It's educating our immune system. It's educating metabolism. It's educating the liver and the fat cells on how big to be and how much energy to store and how much to save. And it's very conserved, because the farmers found that it works in cows, but it also works in chickens. And there's a big swath of vertebrate evolution.
Is there any knowledge about exactly what the microbes are doing that lead to?
There are many things that are going on at the same time. It's changing how the microbes use energy. It's also changing how the microbes are signaling human cells at this critical early time of development.
And there's also a strong correlation between the inflammatory response and many chronic diseases. And so the bacteria are in direct contact with our gut, and they can incite an immune response or inflammatory response. And there's some evidence that reducing that inflammatory response with food or other additives to the diet, including microbes, may be one of the ways we could treat obesity.
I would just add a note of, you might view as slight caution, but actually I think it's incredibly exciting. The idea that these communities, like any of the communities, are amazingly adaptable. So they will recognize conditions, those members and interacting parts of the community that are favored will flourish and the others not. So essentially, these communities remodel themselves under whatever condition they are presented. In this way, they become almost a reinforcing factor for whatever the host or human disease or health state might be. And I think in part that may be why the transfer of a gut community that's now adapted to a state of obesity will bring with it the metabolism and the biology that it took for them to adapt. They bring it to that new host. But it's a piece of the story, and it's a caution in the sense that the microbial community is not the whole story. It could be a reflection of, as well as a driver of, or a reinforcer of whatever the host problem might be.
Yep. Mm-hmm.
And especially with human obesity, there's a lot of complex moving parts there. Even in mice, though, one thing we haven't heard yet about is behavior. And for example, in the toll-like receptor 5 knockout mice that we studied with Andrew Gewitz and Ruth Lay, what was amazing there is that you have a genetic change in the mouse that causes a microbial change that you can transmit from one mouse to another and transmit obesity. And what was going on there was it was almost all behavioral. So the altered microbes made the mice eat more. That's why they became obese. You can make them lean again by just cutting off their food supply to what a normal mouse would eat. And so these connections between the gut and the brain are really exciting, even in the context of things we might think of as more physical, like obesity.
Marty, you said it's not genetic, right? That this phenomenon that we're seeing is not genetic, and yet you have also said that certain conditions like inflammatory bowel disease may actually be transmissible to the next generation. So how does that work if it's not genetic?
Well, when we classically say genetics, we're talking about our human genes passed from generation to generation. But of course, we're also passing our microbial genes from generation to generation. When we're born, it's not quite as precise as the humans, but there's still a lot of inheritance of the microbial genes. And we've done experiments to look at this particular question.
You've done experiments in mice, and this, what are we looking at here?
Yeah, this we're looking at the colon of two mice. On the left side we'll call that the normal, and the right is the perturbed. So the experiment we did here is that we took poop, essentially, from mice that were either normal or had received antibiotics. So we had a perturbed poop and a normal poop. And we gave that to germ-free mice that had no flora, who happened to be pregnant and who also were immunodeficient. They were IL-10 deficient mice. These are mice that spontaneously develop colitis. And so now we gave the poop to the moms, either the normal or the perturbed. Then we asked, would the mom transfer the microbes to her pups? She did. We followed the pups till they were middle-aged. And then when we sacrificed them, we looked at their colon. And what we found is that the mice whose mothers got the perturbed poop, they had much worse inflammation. That's the panel on the right. They had much worse inflammation in their colon than the other. So there was a huge difference. And the point here is that those pups never saw an antibiotic. Their mother never saw an antibiotic. All they saw was a perturbed poop. So this was evidence that the signal, that the disease signal, is being transferred by the microbiome. And it also is evidence that the inheritance is also involving microbiome, involving microbial genes, not host genes.
Right, so not host genes, microbial genes.
Yes.
What are the implications of something like this?
Well, one implication is that many of the diseases that we think are genetic, it may be more than human genetics. For example, inflammatory bowel disease, if you take all of the genetic loci that have been found to be associated with inflammatory bowel disease, that explains 10% of the risk. The other 90% was unexplained. I think this is part of it.
It also has kind of profound social implications, because it means that we can pass our experience on to our children. You know, Lamarck was sort of debunked a long time ago, the concept that we acquire traits during our lifetime and then pass them on.
In other words, if you lift weights a lot, your kids will get big muscles.
Exactly.
Right.
And you know, we don't believe that. That sounds silly. But on the other hand, if we're passing on our microbes and they're a product of our existence and during our lifetime and not just our own genes, then it really has almost I think almost anti-democratic quality to it. That you know, it says that the old adage of pulling ourselves up by our bootstraps and creating our own lives, and every life is a new life and has the fresh start with all the opportunities, just simply isn't true. We are literally inheriting our parents' experience.
Wow. This is a good point to talk about antibiotic resistance. Before we get to that, we want to talk about the bad boy of resistant bugs, one of the baddest boys, and that is C. difficile, which causes untold miserable outcomes in many, many people. And the CDC has termed this a very urgent situation, but there is a very wholesome potential treatment for this, and we're going to talk about it in this video.
I was seeing a urologist for chronic pelvic pain. They were just prescribing me antibiotic after antibiotic.
The antibiotics cleared up the infection causing his pain, but then Ryan began to get different symptoms.
I finally found out that I had C. diff.
C. diff, or C. difficile, is an opportunistic microbe that often afflicts those whose microbiomes have been compromised by antibiotics. It's notoriously difficult to treat and makes day-to-day life miserable.
C. diff can be life-threatening. People can die of this.
None of the medications help with my symptoms. It's just been really draining having this disease for so long.
I have a disease I know is due to biome disruption. Let's fix the biome. Easy to say, challenging to do.
The treatment?
It's called a fecal transplant.
The technical term is FMT, fecal microbiota transplantation.
It's exactly what it sounds like.
Someone with a healthy microbiome has donated his stool to be placed in Ryan's colon.
We drive our camera to the top of a colon, squeeze in some biome, squirt it in with a syringe. That's exactly how elegant or not elegant that is.
Inside of the stool, there are trillions of bacteria that, when transplanted, actually outcompete the C. diff bacteria.
And that repopulates you.
Soon after the procedure, Ryan was free from C. diff.
How does that work, David? Is this more than you bargained for? Is everybody okay?
Well, I mean, the good news is that it's been almost a miracle for people like this patient here and many others. It's remarkable, in fact, that it works, at least in this circumstance. And the idea is that in the wake of this very inflammatory, destructive infection caused by C. difficile and its toxins, the habitat is damaged and yet in some ways unstable and receptive for a new community to replace what is now sort of the ravaged remains of what might be there. And in that particular setting, the provision of a new community, it's really restoration ecology, allows the habitat to now be filled by a community type that really was destined in some ways to be in that environment when the inflammation has subsided. So it's a very particular circumstance, and I think a very important lesson about what we ought to be working towards, not necessarily poop as is, but next generation restoration.
Well, the good news is you can get the poop in a pill.
You can get poop anywhere, actually. It looks like I found that to be true here in New York.
It's so true. It's so true. But should we all be seeking this type of treatment? I mean, can I reverse obesity this way? What are the limits of this right now?
Well, the first thing to say is the C. difficile infection of very specific diseases. We know what causes it. It's acute in onset. The treatment is really has now become the standard of care because it's so good. And it's the proof of principle. But that doesn't mean that other conditions that have been going on for decades, conditions that we don't know the cause of, will be affected. Those are the kinds of questions that have to be addressed in clinical trials. Trials where, just like we've made enormous progress in the treatment of many cancers and the treatment of HIV, that's all come from clinical trials where they've compared one medication to another, one approach to the other, and gradually we've ramped up our approach to these diseases. And that's what has to be done in the microbiome field too.
They are trying it with certain diseases, aren't they? What is the status of that work?
Lots of ongoing trials now for everything from Crohn's disease, ulcerative colitis, to obesity. And any of these conditions that we've been talking about, there are trials underway. I think my own view is that it's going to be a bit tougher and less likely to work easily for the reason that Marty suggested. We don't know the instigating cause or something that's easy enough to knock down temporarily, the way we can knock down C. difficile temporarily, in order to open up that niche space for the new community. A lot of these diseases, as like with health, are associated with intense resilience. The system, whether it be harmful or beneficial, is really stuck in a deep valley that's hard to dislodge. And we're going to have to figure out, I think, ancillary approaches along with the restoration community in order to dislodge, say, a harmful community state and replace it with a beneficial one.
One of the things about the fecal transplant studies that is kind of frustrating is that they work so well with C. diff that patients don't want to stay in the studies. They just get better, or they go out of the hospital after 48 hours and they're done. And so we don't have a good enough understanding of why it works, even in the most extreme case, that we can't really transfer that knowledge to all the other much more complicated conditions that you're talking about.
It's important to point out that C. diff is a disease that is acquired by physicians who give patients lots and lots of antibiotic, kill their natural microbiome, and the C. diff takes over. I mean, so they're very different than some of these chronic diseases.
It can happen even with one course of antibiotics.
It can happen even with one.
It doesn't have to be lots of courses.
But it's an insult that we know that happens, and that's unlike Parkinson's and some of the other diseases you mentioned.
Those are diseases that are going on for years or decades.
Right.
That's the problem. But one of the problems is that all these diseases are heterogeneous. There may be many different subsets in there. And if for example, the fecal transplant worked for 10% of the people, right now we can't really tell who's that 10%. Yeah. And we need much better tools to understand. And for example, back in the old days, breast cancer was considered one disease. And now we know that there are at least 3 major pathways. If we tried to treat everybody with one treatment, we would be undertreating some women and overtreating others. And in kind of a way, that's where we are on the fecal transplant for many of these other conditions.
And even for C. diff, there is a large proportion of patients that respond even to their own fecal transplant being transplanted back. And that doesn't really make sense given the mechanism we think it's acting by. So we don't even understand it with that disease.
Early days.
Yes.
The heterogeneity is even not just with the disease and disease states in individuals and hosts, but also in the therapies. So what is one person's fecal miracle is another person's fecal dud. And because they're just not well characterized, what we need to be working towards is a much more precise blend of organisms and nutrients for those organisms in a particular host, matched in the right way that it makes sense.
Yeah. And the American Gastroenterological Association has set up a new national fecal transplant registry to try to track all this and get the evidence that we need to figure out when it's going to work, what formulation you should use, whose poop is right for you, all that kind of stuff.
Let's move past the colon and up to the brain. For a long time, we thought that these systems had nothing to do with each other. They didn't communicate in any way. But we're beginning to understand that there is a real connection between the brain and the gut. And one of those connections is through neurotransmitters, things that if you are familiar with them at all, you think of them only in terms of the brain, but that's not the case. There's way more of a connection there than we've thought about in the past. Joe, can you?
Well, we knew things that we ignored, and one of the statistics that's so interesting is that serotonin is one of the most important hormones for brain activity and for happiness. And we've known for a long time that 90% of the serotonin receptors are in the gut. Well, what are they doing there? Yeah. We should have been asking this a long time ago because that discovery alone should hint that it's not just about the brain. And so for a long time, we've thought of the gut as the second brain, and that probably is a pretty accurate description. And what they're interacting with, we don't know, but we can imagine they're interacting with microbes and the chemical products of microbes that may mimic neurotransmitters or may produce serotonin itself, or we don't know what else.
We know that depression is a tremendous problem worldwide. It's, I think, the World Health Organization listed as the #1 health threat in the world. There is some work on how we can use this understanding about the microbiome and its effect on depression to help patients.
Again, like many of the things we've talked about, it's a little bit too soon for treatment, but there's clear evidence that the microbiome in depressed people is different from the microbiome on average in not depressed people. And so we need to be asking whether we can do fecal transplants, whether we can change diet, whether we can take probiotics or other chemicals that would change the microbiome in important ways. There was a fascinating mouse experiment where they took the microbiome from depressed people and introduced it into sterile mice, germ-free mice, and they found that the mice that received the depressed people microbiome became depressed.
Wow.
You want to hear about depressed mice? How you know a mouse is depressed? We had a great picture of a depressed mice. I don't know if it's going to come up. There we go.
Aw. Poor little guy. We'll come back to that fella in 1 second, but we learn about the blood-brain barrier in medical school, that nothing gets past the blood-brain barrier. It's a very tight system, but that's not necessarily true where the gut is concerned. They're actually connected in a way. How does that work?
Well, there are a lot of connections. They're just very precise. It's not an easy flow. One of the easiest connections is electronic. So the flow of neural signals from the gut to the brain through the vagus nerve is an enormous route for information to flow. So you could imagine, that's an image of the vagus nerve, you could imagine microbes stimulating the vagus nerve in the gut and that having an effect on the brain. Another way that microbes could be affecting the brain is we know that the composition of the microbiome, in terms of which organisms are there, changes the composition of chemicals in the blood. And the blood certainly can deliver chemicals to the brain. The blood certainly crosses that barrier. So there's no question that there are ways for information to flow from the gut to the brain.
And in the figure that was just up with the vagus nerve, we are hardwired with signals going down from the brain and coming up from the gut.
Is that something that is being studied in humans? Could it work in humans? We talked a little bit about it, but...
That work is just beginning. And of course, we have to remember mice aren't humans, and mouse depression is not identical to human depression, but some of the symptoms are remarkably similar. So one of the tests they use is the swim test, and it turns out that mice will swim for quite a while to try to survive, but if they're depressed, they quit. They just sort of plop over and give up. And so there's the helplessness syndrome, which is one of the symptoms of human depression as well. They also become less adventuresome. And so if you put a mouse on a little island, it will dip its toe in the water like a cat will. But if it's depressed, it will just sit there and hold back. So there are all these symptoms that have corollaries, but of course we're human. We're making a lot of inferences there and imagining what the mouse is feeling. So of course all of this needs to be transferred to humans.
And they called this study Transferring the Blues, which is...
Yes.
Yeah.
The incredibly promising if it can be used to design a useful intervention. The complications, of course, are that depressed people eat differently, their activities are different, the degrees and ways in which they interact with other humans are different, they take medications, and so all of those things we know can independently affect a microbiome. That microbiome, in turn, may now turn into a reflection of all of those factors given to a mouse. May not be so surprising to induce changes that we read as similar. But again, the actual causation and multiple contributions of many factors make this much more complicated, much harder to solve, unfortunately, than that might
Than
suggest.
in the mice, yeah.
So David is emphasizing a very important note of caution. I think we're all here because we're enthusiasts about the microbiome. We recognize that it is a scientific frontier, just like many scientific frontiers, that's going to bear a lot of fruit one day, but maybe not tomorrow, because it's going to take time to translate and to understand all these new findings and to convert them into new approaches to health and treatment. It's not coming tomorrow, except for C. diff.
Right,
but sometimes it takes only one study to take a gigantic step forward. I thought that the brain science that people are doing now with the microbiome would take forever to begin to sort out what's really going on in those mouse brains. And then just last month, a paper came out that showed anatomical changes in the amygdala, which is the part of the brain that controls feeling and depression to some degree. And if you change the microbiome in a mouse, you change the structure of the amygdala. Now that's starting to get to a structural level, and I think ultimately it will be a biochemical level where we can really dissect it. So sometimes it just takes a very innovative study to take that giant next step and get down to the mechanism of what's going on.
It's early times, but would you agree that there are hints that there is a strong association between what's happening in the gut and behavior? Rob, I think you've done some work in PTSD, or you're familiar with this? And
yeah, well, again, this is mostly in mouse models, but with Chris Larry at Boulder and a number of others, we were able to show that if you expose mice to the right kinds of soil bacteria, an organism called Microbiotic vaccae, then you can put it in a social stress condition. So basically, you either put it in with a huge dominant male mouse that would beat it up, or you just let it watch that through glass, which also upsets them, right? And so normally they'll huddle in the corner of the cage. They won't eat, their hair falls out. But if you inoculate them with these soil bacteria, they're very resilient against that stress. And we also did another study looking at children with PTSD, where there were some microbiome differences between those with and without PTSD. But again, we don't have causality in that; we just have association. So the concept that you can modify the mouse's behavior in response to social stress by adding a microbe is very exciting. But it takes a long time to translate that to something that you could do in humans.
And you've even looked at mice with autism, is this correct? And
we haven't, but Sarkis Masmanian at Caltech has done that. And so what is
this we're seeing here?
That's the marble bearing test. So one of the hallmarks of autism is compulsive behavior, and that's a mouse that can't help burying marbles. And the reason
a mouse that likes to bury marbles?
Correct. So the number of marbles it buries in a fixed amount of time is a measure of how much.
Is this like a repetitive behavior?
Exactly, like the mouse version of OCD.
Interesting. Okay.
Yeah. So what happens is when you inject the pregnant female mice with double-stranded RNA, so you simulate a viral attack, and then the pups do that. They have communications deficits, they have cognitive deficits, and they have problems with their GI tract, all of which resemble human autism. And so what Sarkis showed was that there's a molecule called 4-ethylphenyl sulfate that's overproduced in the microbiomes of those pups. And if you inject a normal pup with it, it'll do many of the same things. You can also use a natural microbe out of the human gut as a probiotic to reverse some of those effects. So again, this is all preclinical. There was another consortium of 3 groups in Arizona that did a really exciting clinical trial that was published a little over a year ago with a small number of kids doing fecal transplantation for autism. And so they saw some benefits both in GI dysfunction and also in cognitive function in those children, but it's still very early days, right? It's a small open label trial, and before you believe that it's going to be the next thing like C. diff that can be addressed with fecal transplant, you really want to see a lot more research. But it's very encouraging.
Has anyone looked at populations that have not used intensive antibiotics the way we have in developed countries, and what their microbiomes are like compared to an American's microbiome?
Yeah. So Rob and I have been involved in several studies that have looked at that very question, and the people living out in the jungles, in the bush, who have not had much modernization, their microbiomes are much richer than ours. They're much more diverse. And these are people who live in Africa and South America and New Guinea. They're genetically different. They're on all different diets, but what they share in common is that they have very rich microbiota. And the people who live in Italy, Australia, US, different continents, different ethnicities, et cetera, Japan, but we share modernization and industrialization. Our microbiota are less rich.
Does that have an implication on their health? Are they in terms of the chronic diseases we've talked about today, asthma, obesity, some of the
those comparisons are difficult because we are different from the people living in the jungles in many different ways. But in general, they have less asthma, they have less obesity. But it's hard to prove things from that. But what we can say is that they have a much richer microbiota, and it suggests that theirs is more typical, is more similar to our ancestral microbiota, and that we have lost diversity.
So if I understand this correctly, you're saying that it's possible we cannot recapture that diversity that we most likely started with, like some of the...
Yeah, I don't think we want to say that. We certainly don't want to believe that. We need first, of course, to understand what parts of that ancestral diversity, if you will, are important to us today. And in some ways, of course, you don't want the Hadsas microbiome in order to be able to survive in New York today.
The who?
The Hadza was one of the ancestral, hunter-gatherer populations. That would not be well suited to modern day life, but in turn we ought to be thinking about what elements of it should we attempt to restore, what functional aspects.
Can we restore?
Can we restore and should we restore in order to improve, long-term outcomes and avoid some of the calamities of modern life, if that's
Rob and I are involved in a new foundation that my wife, Maria Gloria Dominguez, is establishing to create a microbiome vault, similar to the seed vault that is in Norway, to preserve all the seeds of humankind.
You guys familiar with the seed vault?
Was that a yes or no?
Yes, they do. They know what it is.
Yeah, so the same idea is to archive those ancestral microbiota now before they're lost, so that at least we have the resource, so that if restoration will be feasible, we will have the materials for the restoration. We don't know yet whether it will be feasible or how. I'm optimistic, but it may take some time to figure out what are the rules, what are the ground rules. Is the restoration of my children going to be the same as the restoration for your children? I think for us, maybe it's a little late.
It's a little late, right?
A little late.
It's not so much the crop trust as the crap trust, if you will, but we really need to preserve this material now while we have the opportunity.
Right. Well, we've spent a lot of time sort of talking about the ways that we've hurt ourselves and our own microbiome over the last decades, if you will, with antibiotics and other things. Let's try to end on a positive note. Let's talk about ways we can enhance or improve or change things going forward. How can we keep our microbiome happy? What should we be doing? Rob, you talk about getting outside. Tell us a little bit about that. Give us some advice. How can we be moving forward for ourselves and our kids, and maybe turn things around a little bit here?
Sure. Well, the most important thing you can do for your gut microbiome, based on what we see in the American Gut Project, is eating a wide range of different plants. So that seems to matter a lot more than if you sit down to a huge plate of kale or if you sometimes eat meat. But that diversity in plant sources seems to be really important.
So, okay. Also, when it comes to particular foods, what types of foods should we be eating?
Well, so at a general level, you should avoid sweet and salty snacks, but I think you knew that already. One of the things that's coming out in the microbiome is that there's amazing individual variability that explains why different people respond so differently to changes in diet. So the science isn't quite there yet to give personalized recommendations, but it's getting pretty close, and in the context of individual research studies, that's working pretty well now.
But don't we know that insoluble fiber, for example, increases butyrate-producing bacteria, and butyrate is a pretty important part of gut health?
Absolutely, and that's really good for most people, but if you have IBD, it's a significant problem, and eating fiber is really bad, so that's why it's always challenging to give generalized recommendations that work for everyone. But there was one Israeli study in particular from Aaron Segal and Aaron Alenev at the Weizmann Institute a couple of years ago, where when they looked at their population, they found for a fairly sizable fraction of people it was actually better for their blood sugar to eat ice cream than it was to eat rice. And they never expected that, right? And when you find that out, you might wonder, is there a test I can do to find out if I'm in the ice cream or the rice category? And the answer is yes, but so far it only really works in Israel if you're eating the Israeli diet. But then the more interesting question is, suppose I'm in the category of people who should eat rice, and I want to be in the category of people who should eat ice cream. Can I change my microbiome to get there? And that's the kind of thing we don't know yet, but it's plausible based on what we can do in mice.
Will we one day be able to match the things that we need to be doing to our microbiome, sort of the way we might other things? In other words, use information about our microbiome to inform what medicines might work best for us?
That's very plausible, and again, in the context of research studies, figuring out if a particular drug, whether it's a painkiller, whether it's a heart drug, whether it's a checkpoint inhibitor for cancer, will work or not based on the microbiome. That works really nicely at a research level. It's not yet ready for clinical use because it has to go through FDA approval and so on, but that's probably coming pretty soon.
That's a huge frontier, because with every medicine that we use, we know that there's tremendous variation in how well it works, what's the level of toxicity, and there's more and more evidence that your microbiome is one of the controlling factors. So if we could understand that, we could make drugs work better. And less toxic, more effective.
Or choose the right drug for the right person.
As well. Yep. As well.
Yeah, and billions of dollars have been spent looking for those answers in the human genome, but they're probably not nearly as much there as they are in the microbiome. And the other thing is that you can't change your human genome, right, to change you from being a non-responder to a responder, but you may be able to change your microbiome.
In just the same way that that variable response to drugs is so individualized and potentially related to the microbiome, so is individual response to environmental chemicals. Heavy metals in the environment, look at lead, look at arsenic. There are huge populations at risk, and yet within a population of identical exposure, there's a lot of variability in outcome, and that's where I think we could have a huge impact in public health on both predicting who is more or less likely to suffer the ill effects of, let's say, lead, because there are genes in our gut microbiome that manage these metals. They've seen them for eons. And if we can predict that, that would be great. And if we could do something about it, it'd be even better.
You mentioned response to different medications, and there is some work in antidepressants. Is that correct, that there's...
My lab is actually starting that work with some collaborators at Wisconsin, so we don't have anything to tell you yet.
What's the goal there, though?
The
goal we all know that for people who are depressed, it might take several trials to find a drug that works for them. There's no one pill that works for everybody all the time.
No, and in fact, the efficacy of antidepressants for any given antidepressant working in any given person is the lowest of any drug group. So it's about a 20% response rate. So typically, people have to go through 4 or 5 drugs to find one that works, and that's a very long and painful and depressing process. And so if we could up front decide just by a microbiome sample which person is likely to respond to which drug, it's a very quick and I think short-term use of microbiome data, because we don't even have to understand the mechanism if, and we hope to, but if we find such a connection, it would just be a predictor that could save physicians and patients a lot of time.
A lot of time.
We already know, for example, that Tylenol response is microbiome driven. So there are differences in the human population. That's been known for years, and there are several other drugs coming along that look like their efficacy is driven, or in some cases toxicity is driven by the microbiome.
Likewise, anti-cancer drugs, the big recent wave in the use of checkpoint inhibitors now is clearly related to the presence of certain kinds of microbes in your gut that either make them more or less effective. It's incredible.
We must discuss probiotics. We've only got 3 minutes, 40 seconds left. Yes or no, Marty?
The future is great for probiotics. We're all thinking that there will be probiotics that people can use. Most of the materials out there for sale in the health food store, grocery store, pharmacy, very variable, almost completely untested.
So caution on the probiotic thing. It's hot right now. It's really hot.
Millions of people take it every day.
Yep.
Astonishingly little data.
Astonishing little data. Okay,
But
Joe?
there are some really good studies that show effects of probiotics. Some of the lactobacilli in particular, Lactobacillus reuteri, which seems to be throughout the mammalian world, which suggests that maybe there's some coevolution of mammals with that bug. That seems to have reproducible effects in people.
But what do you mean by reproducible effects?
That it had, in one case, it had effects on the immune system, and it reduced inflammation. And then there's another study that shows a lactobacillus that reduces depression. That was just one study from England that hasn't been reproduced. So I think there are several things coming along that we will have data, but we just don't have enough now.
But save your money for the moment. What about you, Rob?
Yeah, so just like with drugs, there are studies that particular probiotics work for particular clinical indications, but that doesn't mean that you should just take a whole lot of probiotics all the time, even if you're not sick, any more than you should take a whole lot of drugs all the time, even when you're not sick.
Okay.
There's enough to say that the future is bright. The present is a little bit murky. David Mills at UC Davis did a study and showed that about half of the things that he could buy in a health food store, when sequenced in his lab, were not even the organism that the label said they were. And we know that this, not just the species, but the strain matters. So I think we're working with really imperfect material,
right,
and we can't know yet.
The probiotic manufacturing industry is the Wild West.
The Wild West, okay. Caveat emptor, buyer beware, okay. Marty, I just wanted to end. You have a particular vision for what the future might look like when we've understood the role of the microbiome in our own health. You say that particularly pediatricians are going to have a very different way to examine patients. What's that going to look like?
Well, certainly we're going to live happily ever after.
Okay,
but
good.
Beyond that, I think I envision that the doctor of the future is going to examine babies, and they're going to examine babies' diapers, and they're going to analyze those diapers with the knowledge that is being created in laboratories all over the world, and try to use that information to optimize health. And if there are particular microbes that are missing in that baby that every baby should have, I believe that the doctors of the future will be giving those microbes back, and that there may be specific microbes that that baby is missing that they need, that will become part of the medical care of the future. Not tomorrow, maybe 20 years from now, I'm not sure.
Stay tuned, right?
Stay tuned.
We'll get back to you on that, right? I want to thank the panel here today. David, Rob, Joe, Marty, thank you so much for this incredible discussion.